All right, the next part of this lecture is on dihybrid crosses. And so now we are going to look at crosses between individuals that differ at two different characters. So the dihybrid just indicates two characters. So each character has, again, two pairs of contrasting traits, and this generates a unique F2 generation. So let's see what we need by a dihybrid cross. So again, we start off with a pure breeding population. So we have a smooth green pea, and the genotype of this individual that we've figured out from previous work is big R, little y. So the R controls the round versus wrinkled, and then the Y controls the green versus yellow. So this is homozygous for the dominant allele in the round and homozygous for the recessive allele in the Y. And we've crossed this to a pea plant that produces wrinkled yellow. So you'll notice that there's two different characters that are different. One character is the round wrinkled, so the smoothness, and then the other is the color of it. So we have two different characters that different. And so if we create an F1 individual, we cross this together. So again, there's only one gamete that gets produced. So this individual has to pass on the big R gene allele of the R gene, and it has to pass on the little y. So if we look at the genotype of the gamete, it looks like this, and there's only one possible gamete that it can produce. Same thing for the other individual. And so when we combine these two together to create the F1 generation, we end up with a heterozygote. So this F1 is heterozygote at both genes. This one is round and yellow. So based upon how the cross was done, we have offspring that doesn't share any phenotype with the parent. So it shares the round character with the first parent, but then it shares the yellow character with the second parent, the yellow trait with the second parent. So now we intercross these again, and we create F1s. We cross the F1s. So again, we have to figure out what are the possible gametes that this individual can produce. And so now this individual can actually produce 4 different gametes with equal probability. So it has to give one of the R and one of the Y, but there are 4 different ways it can do that. And I've written those out right here. And so if we do this F1 cross, what we end up are F2s with 4 different phenotypes. So we have parent 1, we have parent 2, we have the F1, and then we have a brand new phenotype, which is wrinkled in green. And so this just kind of gives you an example data that was seen by Mendel, but the ratio, again, is approximately 9:3 to 3:1. So how do we figure out that ratio? And so we are now going to show, so this is, let's see, the cross right here that we will focus on. So this is the same thing, the wrinkled yellow crossed to the green round. And unfortunately, the letters have changed for the color. Sorry, for the color. And so actually for both of them. But again, the gametes that get formed, so we create an F1 that is heterozygote, and then this individual can produce 4 different possible gametes. And so the parent 1, we list, to answer this, we do what's called a Punnett square. You should have seen this before, and we list all 4 possible gametes on the left side of the square. So this is from the first parent, and then from the second parent, we do the same on the top. And so now we simply just combine those gametes together. So this is the genotype of this individual that would be produced if we combine these 2 gametes together, and then so on and so forth. We fill out this entire square. And so many of these genotypes share the same phenotype. So all of these individuals are yellow and round. All of these individuals here are also yellow and round, and then same right here. And some of these boxes also share, for example, the same genotype. So these 2 individuals not only share the same phenotype, but also their genotype is exactly the same. So if we looked at the genotype ratio that occurred from here, we will notice that there are these following genotypes are possible, and then typically we just group these into the phenotypic ratio. So if we add all these together, this is where the 9:3 to 3:1 ratio comes from. And so that's a very important ratio to remember. We're going to see this over and over again in the following chapters. So it's important to remember that an intercross between two heterozygotes that are heterozygote at two genes typically result in a 9:3 to 3:1 ratio. And if we add all these together, you notice the 9 plus 3 plus 3 plus 1 is equal to 16. And then before, when we just had 1, it was a 3:1 ratio, and we added up. So, one thing to remember is with this square is, if this individual produces 4 gametes and this individual produces 4 gametes, and you multiply the 4 times 4 together, that's where you get the 16. And so that's where the ratio kind of comes from. The 9:3 to 3:1 is a ratio of 16 because of that. We could also do a trihybrid cross. So a trihybrid cross would occur between two pure breeding parents that differ at 3 different characters. So it'd have to be something like, if we go back all the way to here, it would have to be something like a smooth green pea that is full is crossed to a yellow wrinkled constricted pod sort of thing. And so when we add that third thing to the trihybrid cross, essentially what we're doing is something like this. So where you have 3 genes and they're different at all 3, and then you end up having a F1 that would be heterozygote at all 3 genes. And so this individual would produce 8 different gametes. So if you did a Punnett square, you would have an 8 by 8 Punnett square, with all possible gametes listed. So I just list the first one right here. And so again, your final ratio is going to be of 64. So 8 times 8 is 64. So this results in a ratio of 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. And so if you add all of those together, you end up with 64. So 27 would share, 27 out of 64 would have all 3 dominant traits. 9 out of 64 would have 2 of the dominant traits, and so there's 3 different possibilities of that. 3 would have 1 dominant trait, and then there'd be 1 out of 64 individuals would have 0 dominant trait. And these are the 8 gametes, for example, that are formed in this way. Finally, the last thing to cover in this class is these ratios, and the work by Mendel assumes that the genes are inherited independently. Or the characters are assorted independently. And so this comes from the fact that the genes he studied were primarily on different chromosomes. And so this, for example, is chromosome 1. So if we're looking at an F1 individual right here, so the genotype of this F1 individual is G little g, W little w. And so if we looked at the chromosomes, it would look something like this. So this G gene is on the first chromosome, and then the W gene is on the second chromosome. And so you get one from the father, one from your mother, and so the big G would be on one of these chromosomes, and then the little g would be on the homologous chromosome. And then the same thing is going on with that. And so because these 2 chromosomes segregate independently during meiosis, you're equally as likely to form the following 2 things. So both of these, this is the first possibility, this is the second possibility, both of these are possible, equally likely to occur, where you segregate the big G with the big W, and then it's equally likely that you segregate the big G with little w. And so what that means is that all 4 of these gametes are equally likely to occur. So if there wasn't independent assortment, if these, two chromosomes for whatever reason liked to segregate together, the blue chromosomes like to segregate together, the result of that would be a skewed ratio. And so it's important this 1/4, 1/4, 1/4 each are equally possible.